I'm Dr. Launen, and this is a short lecture where I'm going to tell you what organisms on the planet have evolved to have the CRISPR-Cas9 system, and what those organisms use it for, what the CRISPR-Cas9 system is, and the basics of how it works, and a little bit of history regarding who first discovered this very important system that now has tremendous applications in genome editing in the scientific community. So I'm going to focus in this lecture, though, on what happens in nature, not on the use of this system for genome engineering by scientists. So let's think a little bit about prokaryotic organisms, those from the domains bacteria and archaea. These organisms are incredibly tiny, and one is shown here. This is a bacterial cell. And these organisms live in an environment where they are surrounded by DNA a lot of the time, especially if they're in places like inside the human gut, where there are a ton of microbes, or on other, densely populated biofilm type surfaces. So these organisms are surrounded by chunks of DNA, and they're also surrounded by viruses. So for every cell, there are typically thought to be up to about 10 different viruses that can attach to and infect that cell. And in prokaryotes, we call those viruses the bacteriophage. In this lecture, I'm just going to say virus to keep it simple. So we're talking about the prokaryotic host cell, shown here in kind of brownish color, and then we're talking about the viruses that can attack those prokaryotic host cells, and they're shown in this green color in this image here. So this prokaryotic cell is just coated with viruses that are attached to and therefore infecting or getting ready to infect this prokaryotic cell. And the question is, how do the prokaryotes survive this sort of infection? They're basically under constant viral attack, and yet they're here, and they've been here for billions of years. So what are they doing to survive that? Well, they have defense systems, and those defense systems are many, and there are probably some that we haven't yet discovered, but they include things like the ability to make restriction enzymes so that they can chop up viral DNA once it gets into the cell, the process of evolution, which allows mutation to all different genetic sequences in a prokaryote, including the ones that encode the receptor proteins that would be found on the cell surface and to which viruses attach. Another really important defense system discovered in the last few decades is the CRISPR Cas system, and that system is what we're going to talk about, or what I'm focusing on in this mini lecture. So these are all ways that viruses can be beaten back by prokaryotic cells. So what is CRISPR Cas? CRISPR Cas is an adaptive, meaning that it can change, immune system, meaning that it can specifically protect. It is found in many prokaryotic cells, and it is a protection system against not just viruses, as I said a moment ago, but also any kind of foreign DNA that's come in, such as plasmid DNA. It's not really known how many prokaryotes have a CRISPR-Cas system, or maybe something like it, but it's thought that maybe as many as half the bacteria that so far have been studied on the planet, and perhaps more of the archaea, contain CRISPR-Cas systems. So this seems to be a pretty abundant or ubiquitous defense system found in prokaryotic cells. What does it look like? Well, if you look at these cells, what the CRISPR-Cas system looks like is a set of DNA sequences that are in the prokaryotic genome. And those DNA sequences, the CRISPR-Cas locus, include the CRISPR part and the Cas part. So CRISPR stands for clustered, regularly interspaced, short, palindromic repeats. They're, in other words, a section of DNA with repeats and spacers. And the word palindromic, or the noun palindrome, means that you've got like a symmetry within those sequences, so that if you were to fold them in half, they would base pair with each other. Cas stands for CRISPR-associated proteins. And so Cas sequences within the CRISPR-Cas locus are the sequences that directly encode a protein, and that protein or proteins can cut DNA, so they're the endonuclease function, and they're also thought to be an RNA processor function. So let's take a little bit of a look at how the CRISPR-Cas works. The reference from which I derived this figure is shown in the slide. So this oval is supposed to be a prokaryotic cell, and here's a virus infecting the prokaryotic cell, and then this virus down here is actually meant to be a virus that's infecting later in the prokaryotic cell life. Okay, but for simplicity, the figure is showing just one prokaryotic cell, and it'd be easy to imagine these two events are happening at the same time, but actually what's happening up here occurs, the acquisition or adaptation phase, and the CRISPR RNA biogenesis phase, and then later, if a subsequent infection occurs, the targeting and clearance phases occur. Okay, so this is a simplified figure, and you really need to think about that. So here is, across the top, the CRISPR-Cas locus, and it would be embedded as a sequence in the circular chromosome contained by this prokaryotic organism. Okay, and then this green line coming out of this virus or bacteriophage is the DNA that is from the virus. So we've got these stages here in the CRISPR-Cas process. So what happens is a virus infects a prokaryotic cell. That's what's happening up here. And then that viral DNA is going to be processed somehow. And the mechanism for that processing is still not really understood. But essentially the processing takes this viral DNA and it chunks it up into discrete regions that are called, or that will become a new spacer, and a new spacer region is shown in here. See these little different colors, right? So those spacers, sorry, pardon the interruption, those spacers are going to be, or that spacer that will have been produced is going to be DNA that is complementary to the DNA of this viral genome. And that's really important, okay? It's a little chunk of DNA that is in the prokaryotic genome that is complementary to the viral DNA that infected this prokaryotic organism. So now you've got that spacer in there, well then what, okay? So then what will happen is that those spacer regions, and each little spacer in here represents a sequence that's complementary to some virus that has previously infected this host, right? In this event, it'll be this virus here, but these other spacers would have come from previous viruses that infected this host, or that infected cells ancestral to this host prokaryotic cell. So that's the acquisition phase, okay? And that's almost comparable to when you or I, as humans, get vaccinated with a vaccine that trains our immune system to recognize some pathogen. This process here, this acquisition stage, is like a vaccination process for prokaryotic cells. And it's called acquisition or adaptation. Okay, so now embedded in the genome of this prokaryotic cell, there's a little sequence that serves as a memory of what this virus looks like. Okay, so whenever this cell replicates, all of this sequence and its associated Cas sequences replicate along with it. So it's maintained in a memory form. All right, so thinking about that, what happens next? Well, next, this spacer DNA will be transcribed. This arrow here is transcription. So this DNA is being converted into RNA through the work of RNA polymerase. So the spacer DNA is transcribed into RNA, and the RNA will be chunked up and modified through a process called CRISPR or cRNA biogenesis. So it's a transcription and modification process. And then what you'll get is, for each of these spacer regions, you will get a little piece of CRISPR RNA. And notice that little hairpin structure? That tells us that there's this internal base pairing happening to allow the CRISPR RNA to take on this very specific three-dimensional shape. What happens next? Okay, we've gone through acquisition and CRISPR RNA biogenesis. Now what happens is you wait, okay? And then if the cell gets infected with the same type of virus, what will happen is the CRISPR RNA will combine or bind to the protein, the Cas nuclease protein, that is produced when you express this gene up here. And that combination of RNA and protein, the RNA is this RNA over here, it's serving as a guide, and the protein is an endonuclease. Together, they are able to find and specifically base pair or target viral DNA that has come into the cell through a subsequent infection. And when they do that, it's a little hard to see in this at this scale, but that red is supposed to be a little X. So what's happening is this CRISPR-Cas complex is cutting this viral DNA. And when the viral DNA is cut up, it destroys it. And when it's destroyed, this virus can no longer infect this prokaryotic cell, and therefore the prokaryotic cell is immune to viral infection. And that's really important. So that's sort of in a nutshell the 3 stages by which a CRISPR-Cas system can protect a prokaryotic cell from viral infection. And you might be wondering why this CRISPR RNA combined with the Cas protein can't together go off and like chop up the DNA up here, since this sequence within the CRISPR RNA is complementary to that spacer sequence. And that's a really good question. And the reason is, in order for the CRISPR-Cas complex to work and to attach to the viral DNA, 2 things have to occur. The CRISPR RNA has to be complementary to the viral DNA, step 1. And step 2 is that complementary viral DNA sequence has to be adjacent to another sequence called a PAM sequence with a little spacer region in between. And that PAM sequence is unique to the viral DNA, and it is not found up here in the CRISPR-Cas locus embedded within the prokaryotic genome. So that's like a pretty cool system, right? Like it shows us that prokaryotes, in a way, invented adaptive immunity. Who figured this stuff out? Many different people contributed to figuring out the CRISPR-Cas system, discovering it, finding it in various prokaryotic cells, and then figuring out like what it was doing in there. And I'm not even talking about applying this to genome editing. I'm just talking about figuring out this system in nature. And so one of the first people to do this was a researcher whose name is Yoshizumi Ishino and his colleagues. And that's a picture of Dr. Ishino shown here. He worked at or works at Osaka University, and in 1987 he published the first CRISPR-Cas sequences discovered, and they were discovered in the bacterium Escherichia coli, or E. coli. Other people worked on this, but another key person was this scientist here and his colleagues from Utrecht University, which is in the Netherlands. And in 2002, Ruud Janssen and his colleagues published a paper where they coined the term CRISPR, and they determined the sequence and function of the Cas genes that were found next to the CRISPR repeat spacer sequences. And they determined that those Cas genes include nucleases, and that you always find these systems where the nuclease sequence is right next to the little repeat spacer sequences called CRISPR. That was in 2002. Then around the early 2000s, I think so it's 2005, Eugene Koonin, who is shown here, and his colleagues, I put this picture in because Eugene Koonin is shown with my favorite virologist, Vincent Racaniello, host to the This Week in Virology podcast series, which, as an aside, I encourage you guys to listen to if you're interested in virology at all. But Eugene Koonin is the one that kind of looked at all this sequence data that existed. And by that, in the early 2000s, more and more scientists were looking at the genomes of different kinds of prokaryotes and seeing these CRISPR-Cas systems. And some people had started noticing that the little repeat sequences were homologous to viral DNA. And Eugene Koonin and his colleagues were the first ones to actually hypothesize that the CRISPR-Cas is an adaptive immune system that facilitates viral clearance in prokaryotic cells. And so Rudolf Barranco and his colleagues, He at that time was working for a Danisco who makes yogurt. They're a sub-company of the much larger entity known as DuPont. So in 2007, they put out a paper that demonstrated that the yogurt-making bacterium Streptococcus thermophilus, which has a CRISPR-Cas system. If the CRISPR-Cas system was present, this bacterium could not be killed by viral attack. And they were solving an applied microbiology question because when you're trying to make yogurt cultures, especially at commercial scale, the last thing you want is for your yogurt-making bacteria to be killed off by viral infection. And so that's why Rudolf Barranco and his colleagues were researching systems to clear viral infection because it had a definitive applied reason or benefit for the food-making industry. So here's a little bit of a timeline taken from a paper put out by Jennifer Doudna and Emmanuel Charpentier. And these are two scientists that I will address in another video lecture. And I'll tell you about how they took the CRISPR-Cas system and they came up with the genius idea of applying it to genome editing. And that's really important. But here I'm just showing you the timeline that they included in the paper cited down here, the CRISPR biology timeline. So there's the discoverer of the CRISPR-Cas sequences. There's the folks figuring out in the early 2000s that CRISPRs contain viral sequences and that identifying the Cas genes. Then Koonin figures out, I think there was an error in my earlier slide, and I said it was 2005 when it was actually 2007, but Eugene Koonin figures out that this is a bacterial immune system. And then there's a lot of work looking at the biology of the CRISPR-Cas system. And you should know that in nature there are actually many different types of CRISPR-Cas systems. And the first one discovered is this so-called type 2 one. So that's a little bit about the history of the discovery of CRISPR-Cas and what it's doing in nature. It's a bacterial and archaeal adaptive immune system, what it is and how it works and how it was figured out. And that concludes this lecture.